Hello, everybody. I got involved in this, as everybody else did, when we sort of got blindsided by Boxwood blight showing up with a vengeance in 2011. Now, initially, I was brought into this as the pathologist and told that this was a difficult disease to control. They'd been using fungicides, and the fungicides were ineffective. And this is what we saw and how we came into this. But this is a very large field that's completely affected by boxwood blight being defoliated. And again, they should all be green. They're going down, and fungicides had been applied. So what happened here? Why weren't we able to control the disease? Well, first of all, I think it had been here longer than we had recognized. The conditions were very good for disease to develop, and it just blew up in our faces. And as a pathologist, when you're trying to control a disease, it's best to control it when it's just starting, not when it's really raging. It's like the analogy I use is trying to put a fire out. If you have a small fire, you can put it out with a little bit of water or an extinguisher, but if your house is raging fire, you're not going to be able to put it out. You can't say water doesn't put out fires. So what happened in 2011, we've been talking, the other people have been talking about the temperature and moisture components that really favor disease. 2011, we had a much higher rainfall than average and lots of days with precipitation, which means we had extended periods with lots of leaf moisture, lots of leaf wetness. So it really did get rolling and really caught us unprepared. So again, now we've got thousands, literally thousands of plants, a huge inoculum source, a raging epidemic, and it's probably no surprise that the fungicides that were applied were not effective. So the result was major losses. So I got brought into this to see, what was going on, how could we try to manage this, what were the other hosts. We inoculated Pachysandra and did work on that. There's been a lot that's already been reported here. My component in this is how do we manage this? How can we use fungicides? Are there some that we can identify, or is there something that we're missing here? So the first thing we did was set up in vitro testing, where we just looked at incorporating fungicides into plant growth media, and then looking at how it affected the pathogen growth, how it affected conidial germination, what's it doing to the fungicides, to the fungus, to the pathogen. And we'd used 20 different active ingredients from 13 different FRAC, fungicide resistance action groups, meaning different modes of action. So we tried to look at a wide range of available fungicides, some which were registered for boxwood and ornamentals, some which were not. And again, we looked at germination of conidia, mycelial growth, and then later on I'll be talking a little bit more about how these fungicides that we found to be effective affected the pathogen in different ways in microsclerotia and other things. Once we looked at that, we then followed up with testing on plants, because obviously the in vitro tests are artificial. They give us an idea of what might be working, but then we needed to see if it worked in the real world. So we did that in greenhouse work, in container nursery areas with 3-gallon pots. And in all of these cases, we inoculated with the pathogen, we gave it as best conditions as we could to get disease going, and then looked to see if we could try to stop it. So initially we had plates, all different. We had thousands of plates, and we went through plates like you couldn't believe, with different concentrations of all these different fungicides, putting the pathogen on and looking at whether it inhibited growth, whether it inhibited germination of the spores. So these are some of the ones that we tested. Typically when I talk about fungicides, I talk about the active ingredients, just because the trade names can be different. There may be multiple trade names for all of these, and they may change over time. But these are the active ingredients: thiophenate methyl, which is Cleary's, and the parentheses number after this is the FRAC group number. So that tells you essentially the mode of action of that particular fungicide, and there are often multiple fungicides with the same mode of action. As you can see, the propiconazole, tebuconazole, triflunomazole, myclobutanol all have FRAC group 3, all different fungicides, slightly different. So in that group, when we looked at now the effective dose response, we looked at the EC85, which means could we inhibit this 85%? A lot of cases that you see people talk about EC50s. Can you cut the growth rate in half? Well, we really wanted to manage this well, so we chose 85. We wanted to hit something that was going to be really effective, not marginally effective. So you'll see that we looked at both germination of Conidia and growth. These particular ones here, no effect on germination, but we were able to find the frac group 1, with, the Clearies and the DMI fungicides, frac group 3s, were very effective at reducing growth of the fungus. Those were the systemic fungicides we tested. The protectant mode, there's a whole range here, lots of different frac groups as well, active ingredient, and also the trade names, or some of the trade names on the side. And in this case, we found some that had effects both on germination and on growth. Frac group 11, Fludeoxonil 12, and then some of the broad spectrum fungicides like EBDCs, the manzate type materials, and chlorothalonil had impacts on germination and on growth. And again, this is the EC85, so we're able to inhibit it by 85%. So what does this mean? Okay, so we can say that we get 3.5 parts per million, and we get to get 85% inhibition. What does that relate to? And is that effective? Well, most of these fungicides are used at rates in a spray tank of about 50 to up to 500. If you're talking about the protectant fungicides, we're using it at over 1,000 to 4,000 parts per million. So if we can get a EC85 of 3.5 or 5, that's pretty good. When you're putting it on at 1,000 parts per million on the surface of the plant, it's going to be much more, and the EC85 is less than 5. Chances are you're going to get pretty good control. So now we moved into where the rubber hits the road, right? Can we put this on plants? Can we get coverage? Can we get effective control other than just on plates? So we initially set this up. We started doing this first in the winter time, so we were working in the greenhouse. We applied on 14-day intervals. Different fungicides, inoculated the pathogen 48 hours after we sprayed, and we gave it a pretty good shot. And then counted the number of lesions 2 weeks after the last spray. Now, in addition to this, we wanted to make sure that we had really good conditions for control. So in the greenhouse, we tented this up. We had high humidity. We gave it at least overnight, so 12 to 24 hours of leaf moisture so that the Conidia we put on germinated and had an opportunity to infect. In the field, we used overhead irrigation, and we gave it a good shot. And in some cases, when it was dry, we actually tented up our field plots to keep it nice and moist. So we're really doing everything possible to do it wrong, to get disease, to try to see if we can control it. So to just kind of summarize this, 4 different experiments. In the greenhouse, we had these materials were effective. Thiophanate methyl, Clearies, that's frac 1. And the frac numbers are important when we go later to talk about how we can mix and match these, because essentially, if everything was in frac group 11, that's still only one mode of action. We're going to have issues with possible fungicide resistance. So propiconazole, fludeoxonil, crisoxamethyl, pyraclostrobin, and chlorothalonil all were effective. And at the same time, we were looking to see if these materials had any phytotoxic effect, and none of the materials that we used that I've used in any of our experiments had phytotoxicity, with the exception that we saw one effect with propiconazole. We got smaller leaves and a little bit tighter growth, sort of a growth regulator effect. And that's not unusual for some of these frac group 3 fungicides. So again, then we also had experiments throughout in our container. We have a little miniature container area. You'll notice the overhead irrigation. And I'll summarize this. We used thiophanate methyl or propiconazole as our systemic. We applied that alone or in combination with protectants. And the protectants we used were pyraclostrobin, fludeoxonil, or chlorothalonil. And we did this 3 times at 2-week intervals, and we continually inoculated, so we had multiple opportunities for infection. And then we counted, at the end of the experiment, 2 weeks afterwards, we counted the number of lesions. And my technicians, I got to give them a lot of credit, they sat and went through all those plants, poked through them all, counting lesions. And then, so this will give you an idea of the percent control that we got. So, no systemics, no protectants, obviously no control, and pretty high levels of disease. We had a series of thiophanate methyl as a systemic in combination with, either alone or in combination with the paraclostrobin, fludeoxanil, and chlorothalonil. Same with the propiconazole. The idea we want to compare which is the better systemic, thiophanate methyl or propiconazole, especially in combination with some of the protectants. Now you have to remember that systemics affect the growth inside, they affect the growth of the fungus, they don't affect the germination at all. The protectants affect germination and sometimes growth, but really it's growth of the germ tube. Once it's in the tissue, the protectants are on the outside. So they complement each other. And you'll see the far column is the percent control. And the final treatment that we had was, instead of using the same treatment 3 times in a row, I alternated. So it was thiophanate methyl and paraclostrobin, then propiconazole and fludeoxanil, and then finally crisoxamethyl and chlorothalonil. So we just kind of did a mix, mix and match and moved it along. And the bottom line is that we were able to get control in the 90s, considering the fact that we were inoculating heavily, given it good conditions for disease. So this was very encouraging. Now, the way we set this up, we could analyze this to see if there were significant differences and do certain comparisons. So fungicides versus untreated, highly significant. We got, the fungicides had a huge impact. When we compared the thiophanate methyl versus the propiconazole as the systemic components, no significant difference. So they both were effective in reducing growth inside the tissue. When we look at using the systemics alone, or whether we use the systemics plus the protectants, obviously the combination of the two was more effective. So you want to get it before it gets in by affecting the germination, and then those that escape, that get in, you can hopefully hit with the systemics. Part of the problem with applying to plants versus doing it in artificial media is just coverage. Physically trying to get in and spray these things, cover every leaf perfectly, is difficult. As you all know, Boxwood have leaves right into the center of the plant. Trying to get complete coverage of that is a difficult thing. And then the combinations versus the doubles actually was still significant. So the bottom treatment where I used alternating fungicides was significantly better. Now the next thing we looked at was, we had questions about, well, how often do we have to spray? Do we have to go out every 2 weeks and spray? Can we extend that spray interval? So we did this again with a sort of a repeat where we did this on 2-week intervals or 3-week intervals. And you look at the number of lesions and the percent leaf drop that we had in here. Actually, this is the percent control of the untreated for number of lesions and leaf drop, where we used protectant fungicides alone or protectant plus systemic at 2-week intervals. We got very good control. 98, 99%. I mean, there's almost no disease. Very little leaf drop. Now when we start getting into a 3-week interval, that falls off. So there's a reason why they're labeled at applying every 14 days is because those materials don't last forever. When they go on, they weather off. The sun hits them. The amount of water we were putting on also had an influence on moving those fungicides off. So they can be effective, but the threshold's low, so we want to get up into those high 90s to try to control this. Now, in addition, we've been looking at screening additional fungicides with chemical companies, some of the things that aren't yet labeled, trying to find other new things that are coming along. Can we get new tools? So some of these we've been looking at, doing comparison with untreated clearies, things like Mural versus Pageant, all significant. So this is Mural is a benzovindofluopyr plus azoxystrobin. Azoxystrobin hasn't been very effective for us, but the other active ingredient is going to be better. Boscalid was not necessarily effective for us, but the paraclostrobin component in Pageant is. 2016, we looked at fluxapyroxad as well, and that also maybe a new material that has some promise for the future. So again, significantly better. I mean, we're talking about 800 leaf and stem lesions versus 2.2 for the BAS 703 at 10 fluid ounces per acre, or 100 gallons, pardon me. And then again in 2017, looked at another series of materials, trifloxystrobin and triimidifon and fluopyram as well, and also with activity. So we're accumulating quite a number of fungicides which have good activity against this particular pathogen. Now, when we were doing, take a step to the side here and say that when we were doing some of this in vitro testing, looking at the ability of the fungicides to inhibit growth, periodically we'd see some of these where we'd get a little bit of growth on a particular plate. So what we ended, and what we wanted to know, I also wanted to know whether the fungicides were inhibiting growth or actually killed the fungus. So what we did here in this little bit was to remove the plugs and then put them on unamended media and see, did the fungus continue to grow or was it killed? So the TM is a thiophenate methyl, cressoxamethyl, tebuconazole, propiconazole, and triflumazole. And the ones that are in red is where it was killed. In other cases, it was inhibited. So it did not grow, but it wasn't killed. Now, the fact that the tebuconazole, propiconazole, and triflumazole resulted in death made me think, well, is it possible they might have some kind of curative activity? Are they going through and actually killing the fungus in addition to just inhibiting its growth? So we looked at this in a couple of different ways. One looking at detached leaves, and where we had a little more control, and then the second on whole plants. So we either did pre-treatment or curative, meaning we inoculated and then treated the leaves to see if we could then kill the fungus that already got in. So we used green velvet, put 200 Conidia on a leaf and a drop. 24, 48, or 96 hours after we inoculated, we essentially dipped it into fungicides at different concentrations, and then looked at whether we got disease. How much infection did it sporulate? And then we re-isolated just to make sure that we knew what we had. So this is what we set up. We turned the leaves upside down, where we got most of our infection on the bottom of the leaf. We put 200 Conidia, and it didn't actually count them out, but we had 200 Conidia per drop and put them on the bottom of these leaves, and then looked to see what happened. Now, in addition, we kind of dissected this and looked at what was going on. Most of the infection went in through the stomates within 24 hours. And this is the fungus that's stained red here. So this is the conidium right here. So this germinates, it produces a little germ tube, goes straight to the stomate and right through. There's another one over here, same deal, grows through, in, and then actually continued on looking for another infection point. Now, we also messed around with testing it on the top, because if it's going through the stomates, what happens when you put it on the top of the leaf where there are no stomates? And one of the things that we found that might be of interest for some of you is that it germinated, it grew, it would infect, it would seem to infect a small number of cells, and then just keep on growing over the top. It kind of grew as though it were powdery mildew for the pathologists in the room. It would grow across the leaf, and if we looked at it under the microscope, in some cases we'd see the strands of hyphae, a couple of spores blown out, and it would just keep growing, but we never got symptoms on the top. So it infected without the stomates as well, direct infection. So going back to our detached leaf assay with the propiconazole. It's a little hard to see here, but take my word for it. These are covered in conidia. These are the untreated. The pre-treateds had no disease. And then as it went through, we got this sort of data. Now, in this case, we had, these are the fungicides, propiconazole, tebuconazole, cressoxamethyl, and thiophenate methyl. Where we pre-treated, now these numbers are the concentration that are different from the control. So when we pre-treated, every one of these was effective. 3 parts per million, we got control. Significantly different from the control. With propiconazole, we got significant differences at 30 and 300 parts per million after 24 hours. It was close after 48, but not significant at 300. The others, no effect. The other way to look at this is, now that was just whether there was infection. In addition, you can't read this, there are 3 different bars on each of these times. This is 3 parts per million, 30 parts per million, 300 parts per million, 24 hours, 48 hours, or 96 hours. And the 3 different bars in sequence are propiconazole, tebuconazole, and cresoxim-methyl. So even at 3 parts per million, at 24 hours, we had a significant reduction in the amount of diseased leaf area. So we still had infection, but it was much less. Even 96 hours after infection, propiconazole was significantly less infected area. So there's something going on. There is some curative activity, it appears to be. Now, in addition to that, so we got some inhibition of infection. We got inhibition of lesion expansion. What happens now when it sporulates to get the next generation? So again, different from the control. All of these inhibited infection and sporulation. And if you don't get infection, you're not going to get sporulation. But the propiconazole and the tebuconazole, these FREC group 3 fungicides, we had impacts on sporulation at 30 and 300 parts per million up to 96 hours after infection. Followed this up with another experiment with the detached leaves where we pulled these out and we just waited to see what happened over time. Eventually they just dried up to the point that we got secondary fungi and things coming in here. But we had sporulation inhibition for the FREC group 3 fungicides that we tested, and it was at least 3 to 4 weeks. After that period of time, keeping them moist so we could get potential sporulation again. We got other things coming in. The cresoxim-methyl, pyraclostrobin, thiophenate methyl, no sporulation inhibition. So it worked on detached leaves. How about with the whole plant? So we inoculated liners with the pathogen. 48 hours later, put on the fungicides and looked to see what happened, then re-isolated. So as you can see, untreated thiophenate methyl and pyraclostrobin, no significant differences, but for propiconazole, instead of 45 lesions, we got 27. So a significant impact. And when we re-isolated, we got less coming back out. So hopefully we were killing it. So what's going on here? And why is it perhaps that we're not killing the entire thing? Well, when we initially did our in vitro testing, we're testing against hyphae as they grow. They're just growing across the plate. But in reality, what happens over time is it doesn't take too long till they start to form chlamydospores in the microsclerotia we've been talking about. And so how quickly does that happen? The chlamydospores form in culture in about 72 hours or less. And that's what I'm calling these swollen hyphae. So in addition to just growing across these little strands of the fungus, they start to get thicker walls, they get like lobes. And those seem not to be as affected by the fungicides. So we actually set some things up where we dug microsclerotia out. I actually had a media, so we didn't damage them, washed them off, exposed them to different fungicides for different periods of time. And bottom line is that only propiconazole was able to kill microsclerotia. So you could actually dip the microsclerotia in thiophenate methyl or cresoxim-methyl or pyraclostrobin at 300 parts per million for 96 hours, and they rinse them off, and they were fine. Propiconazole reduced survival, and then also the colony, they didn't grow well for those that did survive. So again, this is normalized viability, meaning compared to untreated. And this is with propiconazole, so hours of exposure. So let's go to 96, right? So at 3 parts per million, we only got 22% effect. But when we start getting to 30 to 300 parts per million, we were hitting them pretty hard. So propiconazole you got to remember, we're putting it on at rates of about 300. I'm not sure what the concentration in the leaf is going to be. We need to find that out. But it is having some impact, has some potential. So the last thing I want to mention, it was mentioned previously that in Europe they have Henricochet, other things, there's differences in fungicide sensitivity. So we wanted to do a little bit with that too. And as I alluded to previously, when we were doing some of the in vitro testing, we saw some ability to kind of have a little bit of strands here and there. So we actually selected those. Those that tended to put a little more growth out than others, we pulled those aside, we hypho-tipped them, put them on another media, and we did stepwise transfers on higher and higher concentrations. So essentially what we did was, let's say this is the fungus that this isolate, 1194A, we're calling this the wild type. We got growth on 1 part per million, we cut that out, put it on 5 parts per million. If we got growth, we cut that out and put it on 50, and then stepped it up to 250, and that was our selected isolate. So here we are, 1194A, the wild type, what we started with, our selected isolate, and we compared this to an isolate, this CTWH1. This actually came from, it was I don't want to say too much about where it came from, but it was an isolate that came in that we knew had been treated well, it had been sprayed, and we still had disease. So we pulled that out and did a comparison here. And again, these are our EC85 numbers now. So when you see this one, azoxystrobin did not work well for us, period. But, so we looked at crusoxamethyl, paraclostrobin, and trifloxystrobin. And with these are the use rates, so it's going on at, so the crusoxamethyl is 240 parts per million when we spray it, 240 for the paraclostrobin, 150 for the trifloxy. And where it's in red, you can see that it's actually higher, to get the EC85, it's higher than our use rate. So we're actually losing efficacy. We're developing fungicide resistance. So where we selected for growth on paraclostrobin, sure enough, we need to have over 1,000 parts per million now compared to when we started with 0.01. So we've really increased the ability to grow on paraclostrobin. We've demonstrated with small scale that if you continually use the same fungicide over and over again, you are going to select for it. And in addition, when you look at this CTWH1, this isolate that we just got from an area that we knew had been treated extensively with fungicides, higher use rate, higher effective EC85, so 94 instead of 0.01, and over 1,000 for trifloxystrobin. So we have to really be aware of that. So in conclusion, we can control with fungicides. We initially thought that, you know, I was told that this was a lost cause, but it's not. We can get management greater than 95%, which that's pretty darn good for most diseases. Growers would be really extremely happy with 95% control. Obviously, we want to control this to 0. Maybe not practical, but we can get pretty close to it in combination with these other things. The DMI fungicides inhibited sporulation, had some curative activity. So when we start thinking about the sanitation and, you know, moving things out, keep in mind now that if you know what the infected plants or we have conditions conducive for infection, and maybe you can get back in within 24 hours, this might have some activity for you and be a little bit more helpful. And then we also finally demonstrated that these QOY fungicides, the FRAC group 11, we have to be very careful with those because we may have some reduced fungicide activity and we can certainly select for it. We did not see that with the DMIs with our particular isolates. And with that, I will see if we have any questions. And that all fits really into the BMPs. Thank you.